Hello to all the viewers. Today we have the pleasure to hear an interview with Dr. Nikolaos Louros, who is a professor of biophysics at the Medical Center at the University of Texas. And this is a very special interview because Nikolaos has set up a new laboratory at the University of Texas, and he will share the experience of doing it and his choice of putting FIDA into his laboratory with us. Thank you, Nikolaos, for agreeing to be interviewed.
Thanks for having me
here. As I said, we are really curious about your personal experience. So you set up a lab and you could pick many different technologies. Why did you choose FIDA?
Well, good question, but honestly, I had the opportunity to choose between a few instruments to add in the lab, but this was the easiest choice, if I could say. As a recently established lab, I would say that the major challenge that I personally was facing when I was setting it up, and I'm assuming most people in a similar situation would also face, is to make sure that your lab will have the ability to do the various cool things that you're planning to do scientifically when you start. Because resources can be limiting, when starting. So for me, at least, it would be very, very frustrating, if you have all of these interesting things that you're planning to do and you just simply don't have the practical capacity or means to do them. So FIDA was exactly the reason why I wanted to include it in the lab. I think it's an excellent resource for newly established labs because it's a very versatile instrument. So I think that's one of its main properties. It's a signal system that allows you to do, precise measurements like binding of interactions. You can work with different types of molecules, proteins, nucleic acids, even small molecules, which are challenging for other instruments. You can measure affinities, kinetics, stability. You can do quality control. So, and you can do all of these also in near native conditions, which is another challenge for similar other methodologies. And of course, let's be honest, a new lab, cost matters, right? Mm-hmm. So I would say it is a significant initial investment. Of course, it's a new instrument, so there is a cost. It's an investment to get it here, but its operational costs are quite low. It's very easy to use, and it can be very easily integrated in high throughput or medium, at least, throughput workflows, which I think for a newly established lab provides tremendous advantages. So, all put together, I think was an obvious choice.
Sounds like it, indeed. So your lab mainly works with aggregates and amyloid fibrils, right? So now, after you've had FIDA for some time, what are the main applications? What do you do with it at this point?
Yeah. So I mentioned its versatility, right? So we do try to use it for a lot of different things, starting even from quality control of things that we make in the lab. Of course, the nice thing is that it gives you an actual readout. It's not an indirect readout or something like that. So, the hydrodynamic radius is something measurable and something that you can easily understand in your systems.
If I may interrupt you, I'm actually really interested in about your opinion on it, because I know that you have a background in computational science, and with all the developments that we now have with machine learning, artificial intelligence, we need data that is absolute, right? Doesn't matter to you.
Well, actually, that helps me bridge one of the applications of using the instrument. So in our lab, we use a very hybrid approach, using a lot of computational work with the wet lab. So I would say that two of the things that we have used the instrument a lot with using this approach is studying amyloid polymorphism, first of all, which in simple terms is to understand how a protein that forms these pathological aggregates can adopt different shapes, which we now know are also associated to different forms of disease. So understanding how they have this ability to shapeshift is super important for us. both in understanding the mechanisms that drive this process, but also in trying to design molecules to stop the process, potential therapeutics. So we are studying this, and we are doing this through computational design. So we're using a lot of these AI-based tools to do protein design at the moment, and we're trying to see if we can steer polymorphism, in sort of speak.
By changes in the environment?
By changes in the sequence of the proteins.
Okay. Mm-hmm.
So we are developing different constructs, and then we express them and want to see if they actually form the stuff that we are computationally thinking that they are. So having the ability to measure hydrodynamic radius, for instance, is a very quick first step to measure and see if what we designed computationally actually falls the way that we expect it to. In our experiments. And then we use a similar approach where we try to design molecules to bind to these aggregates specifically. And these things have potential therapeutic potential. The challenging part with our lab is we do a lot of peptide-based work. So we have a lot of small-sized molecules as binders, and the aggregates tend to be really large in size. A lot of traditional binding approaches are very challenging to measure these kind of interactions, whereas FIDA has a very high sensitivity. It can provide you changes close to the nanometer level for binding interactions. So it's helped us a lot in trying to measure such interactions.
Okay, lovely. And what about kinetics? You've used it a little bit. Do you have an opinion?
It's very straightforward, albeit a bit indirect, of course. So, it's a bit challenging because pretty much they're independent experiments that you have to do. You have to have independent curves in this case, but you do get decent results out of it. So for some of the binder designs that I have mentioned, of course, we have also used the kinetics approach of it. And we're also collaborating with other labs, both here at UT Southwestern and outside, who have also developed specific antibodies or nanobodies, as binders for their targets. So we very frequently use the kinetics module for those kind of applications, which is very important.
Okay, lovely. And now I'm wondering about the practicality of speed, of assay development. When you have set up a new lab, I'm sure it's a lot of work, and you have to get your processes in place. How was it for you to develop all the assays that you're performing?
Yeah, it's always challenging to develop new assays, I would say, but actually FIDA was quite straightforward. It's super easy to use, which is again a big benefit for a new lab, because you have new people. You also have to be involved yourself. You're more actually hands-on involved in these kind of experiments at those steps. So it helps a lot to have a system that can be easily shown and taught to new people, and then they can pick it up and use it themselves very quickly. So in terms of that, that's very, very helpful. I would also say that because the readout is quite simple of the samples, it's also quite simple for people who understand it to try and utilize it in different ways to develop, new types of assays. So I think that's been very, very interesting for us, and we're trying to challenge the instrument a bit.
Okay, I'm curious about that. So what other applications did you develop?
So one of the things that we are trying now, and I think this is where it can potentially stand out a bit, is to try and measure binding interactions within complex matrices. Because that's always been a very challenging approach with other methods. So we are some of these binders that we are developing in the lab, trying to see if we can if they have any potential diagnostic potential against patient samples, for instance, extracts from patients directly, which are in complex solutions, pretty much. So we'll see how that goes. Hopefully soon you will have some information on that stuff.
It sounds very, very impactful. Of course, all of the research, gets us closer into to knowing more about the diseases and finding the right ways of treating them. But this sounds as a you're working so close to the patient. So you work on what kind of raw samples in that case?
We've tried from brain extracts, from various other liquids like bloods or serum and so on. We've tried CSF in certain cases, just some of the samples that we have tried. And of course, a lot of in vitro produced materials, purified proteins, synthetic peptides, even some small compounds. We have tried a few things already in the lab.
Yeah. And that worked well. So the flexibility of the instrument when it comes to working with different types of samples, that works for you?
It is flexible, but of course, depending on the samples that you have to change a bit the protocols of the experiments, right? Some of the samples are a bit more cloggy or a bit more sticky, or so you need some to plan in some washes or adapt your protocol a bit more depending on the samples that you're using. So that's part of the process, but it's manageable for sure.
I hope that the quality control dashboard is helpful in that.
Definitely.
Okay, lovely. All right. Is there anything else that comes to your mind when you're thinking FIDA? Is there any way you're incorporating it in the processes currently at the lab?
Well, we do the binding interactions that I mentioned. We do a lot of. We also measure for the different polymorphs that I mentioned earlier. We do some stability analysis for those fibrils, both chemical, thermal denaturation of the aggregates, because it's important when you design a new fibril to see if you've improved it or destabilized it or, how you affect it pretty much. So we've done some of those assays. And there are several external collaborators. I should also mention this is probably another reason why FIDA could be an attractive thing to add in a new lab, because it helps with setting up a lot of external collaborators that have an interest in using it. It's a very easy and quick to use instrument, so that's why it has an appeal to externals. So we have actually used it for various different types of projects, but of course those are not directly associated projects to our lab. But the point is, it has been used for various different approaches.
Lovely. I'm very happy to hear that there is such a big contribution into the versatility of your laboratory. Fantastic. Thank you so much for the interview. I wish you best of luck for developing new assays and for having new publications in. And whenever we can support you, you know who to reach out to. Thank you so much.
Thank you for having me. Thanks.